Okay, so Jean says, whatever you wish, enjoy all your sharing. Thank you, Jean. So why not we do this? Let's discuss this. Although if you look at the comments yesterday under the study, that was such an interesting set of comments. Um, some of the commenters thought that the study itself was invalid because of the explanations in there. Some thought it was just a garbage study. It amazes me to observe that some researchers who worked for 13 years or more to put together the first study in the world that showed how anxiety impacts the brain tissue, which tells us that we need to control our anxiety whichever way we can. And then we feel comfortable enough just to write a comment saying, this is a garbage study. It just amazes me that such assessments are done in such a careless way. So with this, I'm just going to look at where my pen is and we would start. So just one quick second. Do you see that Luffy is sitting here? And it is so strange that my pen is not here to draw. Here it is. All right. Thank you, Jean. All right. So here is the important thing. Let's do a quick review of the study. The study said, and this is a 13 years or more long study. Remember that studies, if the duration of the study is 13 years, then the actual study is not 13, it is more than that, because first they have to plan for the study. For that, they have to study other studies to understand that has some other study covered this hypothesis. Then they have to plan, go to the ethics board, get the approval for the study, then start doing the study, do the follow-ups, keep the records, keep them processed. Then at the end of the study, pick up all of those records and reprocess them and run them through various algorithms to find the results for us. And how callous of us to just sit down and say, well, this is a garbage study. So with this, let's discuss the mechanism. The basic problem of the brain tissue impact, and I would actually add cardiovascular system impact from anxiety, is because of three primary reasons. One is that there is stress hormone, corticosterone, that are released more, and that has an impact on the brain and cardiovascular system. The second one is sympathetic system goes in overdrive, releasing more norepinephrine and epinephrine, which then causes the damage to the blood vessels and heart. That damage to blood vessels is reflected by damage to the brain tissue. And the third one is the habits. When somebody is anxious, when somebody is depressed, when somebody is sad, their habits change. They would not care much for how well they are eating, how good food they're eating or not. They probably start smoking. They'll start some other habits which are just a reflection of their state of mind. They're not in a healthy state of mind, and it is just a reflection of that. So that those habits, unhealthy habits, would then as well reflect into body's inflammatory state and blood vascular issues. So this is the basic idea. So let me now explain how this happens. Dr. Turab Sayed is here. Dr. Turab, welcome. Drbeen, consultant orthopedics, stress and anxiety leads to highest risk of in surgical patients, CRPS in surgical undergoing limb surgery. And it is very true. The thing is this, that this anxiety causing effect on the heart and blood vessels is known. This is the first study. We should be grateful to these researchers who brought in, for I'm sure, at least 14 years of their work to tell us that, hey, take care of your anxiety. How do you take care of your anxiety? Everybody would take care of that differently. As I said before in my Patreon talk, somebody would like to listen to music. somebody would like to get psychotherapy or psychiatric medicines, or going out for a walk, or sitting down with their friends, laughing, um, and at the end of the day, the basic problem that causes anxiety need to be fixed as well. So whatever is the way that would help you, but that has to be done. So um, let's look at the mechanisms possible. Now, these mechanisms are not written in that manuscript. This is my improvisation for explaining the mechanisms. They had one paragraph about the mechanism, and I'm going to elaborate that. That means there can be inaccuracies, there can be irrelevance to the study, and we can then discard it. I'm going to hide this video anyways because this is an improvised video. So I'll explain it, then I'll let you watch it for another few hours, and then I'll hide it. So let's start. So here is how it happens. A person who's anxious, the first thing, I mean, there are many, many effects of anxiety. I am going to stay within the context of this particular study and the brain tissue impact. So the anxious person will have hypothalamus, pituitary, and adrenal are suprarenal glands that are sitting on top of the kidneys, adrenal axis. This axis becomes active. And this happens in all kinds of stresses, not just anxiety. Anxiety is a reflection of the stress, right? An uncomfortable feeling that disrupts our life, that is anxiety. So this stress hormone is released every time we are under stress. So what happens is our personality areas, our system in the brain, our limbic system, when they assess that we are under stress, they activate the hypothalamus pituitary axis, and hypothalamus sends a message to pituitary to send that message through corticotropic releasing hormone or CRH. This hormone goes and acts on the anterior pituitary, which then releases a hormone called ACTH, adrenocorticotropic hormone, that goes and act on the adrenal glands. So stress caused the hypothalamic region to become activated to release CRH. The CRH went to anterior pituitary, and that pituitary would in turn release ACTH. ACTH would go to the adrenal system and ask those cells to release corticosterone. So the result is that we will release corticosterones. Now, corticosterone have a lot of effects. Glucocorticoids, for example, lots of effects. I'm going to just pick and choose some of the effects that are relevant here, or that are most relevant here. So the first effect in our brain, sorry for this crooked brain, but let's just live with it. So in our brain, there is a tissue called hippocampus. Hippocampus is the gateway for memories. When we have new memories, so imagine that right now you are listening to this lecture. Some of you are continuing to listen because you thought maybe this is interesting. Some of you are going to say, you know what, not interested. Some of you are going to say, well, this is a good lecture. Or like yesterday's comment, some of you are going to say, not very good. So what happens is that when whatever event we have, whatever sensory input we get, and we get sensory input, for example, from our eyes, from our ears, from our olfaction or the smell, from touch, pain, pressure, temperature, from our body, then we get the sensory input from our muscle tendons that are telling the brain for how much is the pressure on the tendons, and the muscles would send their tension as well, and that would tell us what is our posture and how to maintain that posture and so on. So the higher centers, that is brain, is bombarded with a number of sensory inputs. And the brain has to actually go through all of that input to figure out what is relevant and what is not relevant, what is good, what is bad, what is neutral. So right now I'm sitting here at this desk and I have my speakers over here and the audio system and the video system and the camera over here and the monitor over here. I am not focusing on them at all. But from my visual system, all of that information is actually entering my brain. So what happens is that the brain tissue's job is to sort those out, to discard some of them, to say this is not relevant, this is not important at this time, and then take the remaining things, whatever is good or bad, and try to remember that. If you are going to your office, many times you will start driving from your home to your office and you would not even know that you reached your office. Why? Because your sensory processing systems have been discarding the information that you were collecting on the way towards your office. On the other hand, if there is an accident on the way and you let's say you were there at a time when the accident just happened and you saw someone in trouble, you would continue to remember that because your limbic system would declare that event to be painful, that event to be concerning. This would not be a neutral event. This would not be whatever event. So you would come across happy events, or you would come across sad events or negative events, and you'll come across neutral events. When you come across positive or negative events, they are routed to the hippocampus and asked to be remembered. So when you have to remember something, so memory is such a large concept, and there are so many hypotheses that I cannot actually sit here and tell us how memory becomes formed. There are many hypotheses. I'll just take one hypothesis and explain how that works. So now imagine that the memory or the event is injected into the hippocampus, and hippocampus's job is to now integrate that memory into the remaining events and into our brain's personality parts. So what happens is, let's say this is a neuron that is involved in that memory. So every kind of event has a different set of neurons activated. Not the whole brain doesn't become activated. If the whole brain becomes activated, we'll get epileptic attack. So some parts of our brains will become activated relevant to that particular event and the sensory input from that event. So imagine that this neuron became active. This neuron is now going to send messages to other neurons that are also relevant to this particular situation. So how does this work? We know that the electrical activity from a neuron will then pass through a synapse. A synapse is where one neuron is giving the information to another neuron, and the electrical activity is converted into chemical activity in here, then electrical activity again. So, and I've discussed this on Drbeen. There is a lecture on pain management and some of the brain tracks and how do they work. I've discussed this in detail over there. But generally, what would happen is that the neurotransmitter will be released from one neuron, and there are receptors on the other neuron that would receive that neurotransmitter, and this neuron in turn will either become activated or inhibited depending upon what is the neurotransmitter that is released. When this particular event occurred, some negative event or a positive event, your brain system tissue under the guidance of hippocampus then will start rehearsing it. This is why when you have an event, you start remembering it again and again throughout the day. And every time you remember it, it is kind of with the same intensity, as if you're reliving it. What is happening? Your brain is trying to memorize this. Your brain is trying to convert that from a short-term memory into a long-term memory. And one part of that is the neuronal or synaptic transmissions. So what happens is when a neuron is firing again and again and again, it will actually create physical changes in this neuron. The machinery that makes the neurotransmitter, there will be more of that machinery made in that neuron, so that neuron can now put out more neurotransmitters. Similarly, on the other side, the receiver side, who's receiving the bombardment of the neurotransmitters will then have to decide that, oh, I'm so sorry. The receiver that is receiving this bombardment of the neurotransmitter would say, you know what, I need to make some more receptors to be able to capture this, all the information coming to me. Or it could decide to downregulate the receptors. The point is that there is going to be a change in the actual physical structures of this synaptic area. This is called facilitation. Facilitation, if I am spelling it correctly. Facilitation is one of the mechanisms for memory. We actually do not really know very well how the memories are formed. So now if I go back here to the hippocampus is the gateway to the newer memories. And imagine now another very important thing about hippocampus, and that is that in the hippocampus, we form about 500 new neurons every day. Neurogenesis occurs in hippocampus. There are stem cells that sit in the hippocampus to make new neurons, 500 new neurons every day. And then, of course, 500 neurons die as well, so that the volume of the hippocampus is maintained. And it is such an interesting thing that by the age 50, all of the hippocampal tissue has become recycled. With those 500 new neurons every day cycling and 500 dying, by the age 50, we have a new hippocampus, and that would continue. The corticosteroids have a very interesting effect on the hippocampus. Corticosteroids have effects on the whole brain tissue, and one of the important effect is the apoptosis of the brain tissue, or the death of the neurons. But here, what is interesting is that the corticosteroid in the hippocampus work on the hippocampal stem cells. Remember, the stem cells are the cells that can make their own type, they can replenish their own community, plus they can make some other type. That is why they call stem cells. The stem cells that are going to make new neurons for hippocampus, when they are acted upon by corticosteroids, their function is reduced. So new neuron production will reduce. Tell me that this is a garbage thing to talk about. I became just so annoyed with this kind of a comment. Anyways, so these stem cells reduce their function. The result is that we are not making as many new neurons as we should. However, the neurons that are dying, they continue to die. So now, if you think about it, net in the hippocampal tissue, more tissue is dying and less tissue is being produced because there is stress hormone that is present in the system. The result will be over time, and according to that study, people who had their anxiety resolved within 5 years' time, they had zero effect. That means the damage for 5 years, even if it was occurring lesser than 5 years, it was not resulting in dementia. But when the damage continues to occur longer than that, then this problem of hippocampal atrophy means reduction in size, because the cells are now going away, that atrophy actually now starts leading to cognitive decline, leading to dementia, leading to problems with memory. Did you ever notice that when you're stressed, you start forgetting things, you start having problem with the memory, you start doing word searching? So these are temporary effects. However, when the stress continues, that would over 5+ years, according to that study, there may be more studies in the future that would come back with the precise bracket of years. But according to that study, 5+ years is when there is enough damage done that cognitive decline appears. So this is just one of the issues. The second issue, corticosteroids, especially in the hippocampal tissues, reduce the glucose utility, and the glucose metabolism is disrupted, which would also mean that we have now less nutrition available to the hippocampal tissue, which will mean the set of problems that would occur, that when the cells, the neuron, have less nutrition available, what will they do? Number one, they would reduce their action. So their activity will go down. That would, of course, mean memory issues and other emotional issues. Secondly, if you reduce the energy production in a cell that is the mitochondria are working lesser, but the demand for the energy of the cell is more, then the mitochondria would start becoming disrupted. What would they do? A mitochondria would try to divide into two to increase the volume of the mitochondria present and hope that each of them is going to grow with more nutrition in them and with more machinery in them and try to catch up. But if you're already giving them less nutrition, and I'll come to that why are we giving them less nutrition? But if they have less nutrition, if they have glucose utility that has become disrupted, these mitochondria would just keep doing the fission and continue to become disrupted. So mitochondrial damage would start appearing as well. Apoptosis. Apoptosis is a graceful cell death. A cell can go through many processes of dying. One process of dying is autophagy. Autophagy in a cell can be of two types, destructive or constructive. Constructive autophagy is when a cell can pick up the trash material present in it, break it down, use it to make ATP and work with that ATP and use that for its advantage, or recycle the material that is trapped in this trash to make other things, enzymes. Destructive autophagy is when the cell is going to start eating up its own organelle, for example, the Golgi apparatuses or endoplasmic reticulums or other structures that are present. And when the cell starts tearing down those structures, the cell is going to die. So autophagy is one process where the cell death can occur. Apoptosis is another process where a graceful cell death occurs and inflammation does not happen as a result of apoptosis, but man, we lost a neuron. So even if we say that the cell death or the autophagy, the apoptosis was constructive, it is non-inflammatory, we still have a problem that a neuron went away, right? A cell can die by inflammasome or pyroptotic pathway where it causes inflammation. A cell can die by ferroptosis as well, where there would be inflammation. So there are many ways that cell can die. Cell can also enter a survival mode as well, where it would become exhausted, become inflammatory, and not do proper functioning. So many fates of a cell. I actually did a lecture called Cell States, Stressor States and Fates. It's on drbeen.com. The problem is the neuron would start dying. This is just one effect of corticosteroids. And this is not just on the hippocampus, other brain tissues would have this effect as well. So again, the solution for us is to figure out how do we keep our stresses under control. Now, just 1 second. There was someone who was disrupting too much. I just let them out. Okay. Now, the second thing, other than the steroids, and I'm not talking about the steroid action within the body, it would just become a 10 hours long lecture. I'm going to go to the second part of the stress causing sympathetic system overdrive. So stress also causes sympathetic system to start working at a higher drive. What does that do? That causes more epinephrine and norepinephrine to be released in the system. Now, epinephrine and norepinephrine have a huge impact on the blood vessels, on the health of the blood vessels, and on the tissue around the blood vessels. So what happens is the following. Epinephrine and norepinephrine's job is to reduce, they do many things, but one of them is that they cause blood vessel narrowing. They cause vasoconstriction. Now, vasoconstriction, if needed, will be the right thing. For example, if we are maintaining our blood pressure and we are under a fight and flight mode, we are under some kind of a stress where we need to run and we need to do some physical activity, then we need to have our blood pressure raised. And for that, vasoconstriction is a helpful thing. On the other hand, if we are just sitting, if I'm sitting here at my chair and I'm stressed, I'm not fighting any lion over here. I am just sitting here and the fight is in my head, causing stress and causing sympathetic overdrive because the sympathetic system doesn't know that I'm actually fighting something or not. So the sympathetic system is going to start producing norepinephrine and epinephrine. They would cause the narrowing of the blood vessel, which is intended to increase the blood pressure. But in a healthy individual who is not really in a need for doing this, they would develop hypertension because there is a continuous narrowing or vasoconstriction that is present in their system, the person who is anxious. Now, not only the vasoconstriction is present, which will result in hypertension, and think about it, hypertension itself would then start causing cardiovascular damage, the heart damage, because it would put load on the heart. But before we go over there, the other thing that norepinephrine and epinephrine do is that they activate the endothelial cells of the blood vessels. So what happens is that when the norepinephrine and epinephrine work on the endothelium, number one, they increase reactive oxygen species production. Why do they increase the production of reactive oxygen species? Because think about it, why was the norepinephrine and epinephrine released? For stress. The stress could be a pathogen. The stress could be a virus or a bacteria or a fungus or a worm or something. Stress could be some damaged tissue. So when these events occur, our body need to produce more reactive oxygen species to kind of destroy these bad things. So generally, the cells start increasing the production of reactive oxygen species. So now imagine this person is under stress. He's not fighting any infection. He or she is not fighting any infection or any virus or whatever. And their reactive oxygen species within the endothelium is increased. What will that do? Reactive oxygen species are going to start causing damage and inflammation would start. For us is this association with anxiety and dementia new knowledge, and what are the repercussions with regard to general practice doctoring? So very important question for us. If you look at yesterday's discussion, they did say that there were previous studies that had tried to understand the association between anxiety and dementia. There is already, we know that anxiety and cardiovascular system impact is known. We all know this thing. And there are many, many studies. However, dementia studies, according to the manuscript yesterday, and I would invite you to read that once, in there they said that previous studies were non-conclusive. And the reason for that was that these were not longitudinal study. If you have somebody under stress, and then the next week you test them for dementia and you say, well, they have stress, but there is no dementia, so fine, there is no problem. In this study, they looked at it longitudinally over 13 years, up to 13 years, I should say. And because of that, this is the first kind of a study which came back to say we actually observed people under stress for years and years and years. And then we saw if they would develop dementia. We compared them to those who did not have stress, and we saw what is happening with their dementia or not. And we found significant increase in the individuals with continuous stress and anxiety and their brain tissue impact. So to answer your question in a short way, yes, from the nature of the study, this is the first type of a study. Now, the repercussions for the practice doctoring. Two things here. Such a beautiful question. They say it in their manuscript that for the first time, this study shows that anxiety can be a modifiable risk factor for dementia. That means if you find a patient with anxiety, make sure that you try to manage it because it is a modifiable risk factor for dementia. So that means if we don't manage it, cognitive decline can start occurring. And fortunate for us that if the anxiety is resolved within 5 years or lesser than 5 years, then the effect on the brain tissue is zero. So that is the good news. That means we have some years to manage the anxiety. So that is one. And the second is, of course, anxiety management should become a more important factor for doctors because it can impact not only cardiovascular system, but it can impact the brain tissue as well. So Vijay says, I guess I'm screwed. So no, not that. Look, if somebody was anxious and the anxiety got resolved within 5 years, there was zero effect. Number one. Number two, if I am anxious and there is some impact on my tissue because I stayed anxious for 8 years or 9 years and now I have some negative outcome, I still need to manage it so that there is no further outcome. And maybe that would give the brain a chance to start recovering as well. So nobody's screwed. It says that this is important information for us to know so we can manage ourselves better, so we can be better healthy. Why not? I was just completely blown away by people trying to badmouth this study and shun it. When somebody is coming to table saying, here is a very simple thing that we can look at for our well-being. Okay, so reactive oxygen species are increased. That is one. Secondly, the nitric oxide production from the endothelium is reduced. Now, endothelium produces nitric oxide for vasodilatation. What happens is that the blood vessels, arteries, especially large and medium-sized arteries, not capillaries, they have muscles like a sleeve around them. These muscles can relax to increase the diameter of the blood vessel, or they can contract to reduce the diameter of the blood vessel. And that is how blood flow, blood pressure is regulated. So I'm looking at some comments here. Okay, so some side discussion is happening. So nitric oxide production is reduced under the effect of these hormones as well. Let me just call them stress hormones from now on: norepinephrine, epinephrine, cortisols. When the nitric oxide is released, the job of the nitric oxide is that it is released from a cell, endothelial cell. It then diffuses outwards into the blood vessel wall, intermediate wall, which is made up of the muscles, and then it causes relaxation of the muscle. When the muscle is relaxed, the diameter of the blood vessel is increased, blood flow improves to the downstream tissue, and the tissue becomes happy. It gets more nutrition, more oxygen, it gets more waste product washed away and more carbon dioxide washed away. So it becomes generally healthier and fresher and better. Now imagine, so let me talk about one more thing. There is a concept of auto regulation of blood flow to a tissue. That means, for example, right now, if you're sitting here, you start moving your biceps, you start exercising, what would happen is that movement of the muscles will make those muscles extract more blood towards them. And that is called autoregulation. They would selectively increase the diameter of the blood vessels that are supplying them, instead of increasing the blood flow to every part of our body. So this is called autoregulation. Whatever tissue is working more gets more blood flow. Now imagine that the nitric oxide is not being produced correctly. Now a tissue needs to autoregulate the blood flow to itself, and it will not get the correct blood supply. Imagine that somebody is under stress and they are working on something. Let's say they're doing some hard brain activity. Imagine they're planning something. Imagine they're preparing for an exam. Whatever they're doing, a lot of brain activity is needed. That means more supply of the blood flow is needed to their brain. And here comes the nitric oxide, which is stalling or hampering that supply, that increased supply. So that is, of course, a negative outcome. Now, when the blood vessels stay constricted, they would put pressure. That this is called increased total peripheral resistance, because the blood vessels are narrowed, because there is vasoconstriction, that causes hypertension. Plus, that puts a load on the heart as well, and heart has to now pump against this narrowed system of the blood vessels, and there is a pressure on the heart now, which would then cause heart to start becoming remodeled if this is consistent or chronic. Just like a bodybuilder's muscles become different because of the exercise they do, similarly, heart starts becoming different as well, and that is called cardiac remodeling. And the cardiac remodeling can include the muscle hypertrophy. The muscle can become large. The muscle geometry can become different. The cardiac muscles are arranged in a way, in a spiral way, that they not only um, reduce the diameter of the heart when they are beating, but they also shrink the length of the heart as well. And so when these muscles start rearranging themselves, and when the remodeling of the geometric structure is done, then the heart becomes less efficient. Then there is electrical activity of the heart, and that system becomes different as well. So there are many changes that comes to heart, which can actually make that heart prone to have an issue later on in the life. But I want to continue to focus on the brain side. So when the blood vessels are narrowed, when they're not working correctly, they will not be able to supply the brain tissue very correctly as well. So that would cause an issue. Okay, so Faraz says, what is the main mediator of morbidity of all the ones you have mentioned, or is it undetermined? What would taking nitrates help, or arginine, citrulline? Yes. So I'm not going to go into the depth of this answer, but yes, taking nitric oxides at that time will help. Okay, so back here. So now, how many ways the brain tissue is under stress as a result of stress and anxiety? One is that the corticosteroids are causing an issue with the brain tissue. Apoptosis is occurring. Some new cell formation is hampered. The neurogenesis is hampered. Brain's electrical activity is hampered. Brain's metabolism, glucose metabolism is hampered. That would create an effect. Then norepinephrine and epinephrine would compromise the blood flow to the tissues, especially when these tissues are actively working and they need more blood flow, and that would cause an issue. Then would come the psychological outcome, and that is the person becoming more less healthy in their habits. And imagine if we start eating bad food, let's say more carbs or foods that can cause more inflammation in us, that would in turn have effect not on our body, that we will start moving towards diabetes and other organ damages, but blood vascular damage would cause hypertension. Heart damage will make us prone to have coronary artery problems. Heart inefficiency would then lead towards the possibility of strokes. Now, going back here, one last topic, and then we stop. And then I would hide this video in another hour or so. The last topic that I want to discuss is the endothelium activation under this norepinephrine and epinephrine doing another interesting problem. And that is activated endothelium will start expressing the cell adhesion molecules, adhesion molecules. These are also called CAM, cell adhesion molecule, CAM. The CAM, their job is to try to stop the flowing blood cells and capture them in that area to help the endothelium produce inflammation. So endothelium doesn't know that why do I have the stress hormones running around. All it knows is that the body is in a need for inflammation or for stress management. So when that is consistently present, endothelial cell adhesion molecule would increase. That would trap more and more lymphocytes over there. That would cause local inflammation. Another important thing is that when we have reactive oxygen species and other chemical substances released by endothelium, they would cause platelet activation, and now the thrombosis would start, which would further compromise the blood supply to various tissues. So I have totally bypassed the effect on the heart because that's another 1 hour long lecture, but you know that a person under stress would have palpitations, would have a different heart rate, even paroxysmal attacks of palpitations that could mean arrhythmias can occur, atrial arrhythmias can occur, then ventricular arrhythmias can occur, even the cardiovascular blood supply gets compromised and a heart attack can occur. So we know those things from our past researches. The new thing that was this study presenting to us was that such events, when they happen to brain, then they lead to brain tissue damage, which then leads to cognitive decline or dementia. And the time frame they said for dementia to occur was 10 years from the anxiety. And again, important thing to keep in mind, lesser than anxiety resolved within lesser than 5 years did not have any effect. But anxiety that continued beyond 5 years, that had an effect. So this is the discussion. Thank you very much. I just improvised it. I'm going to leave it up for a couple of more hours and then I'm going to hide it. If somebody has a link, they can watch it, but I'll just hide it afterwards because this is an improvised one, not great drawings, just doodling to discuss this. So Helen B says, how about caffeine? Yeah, caffeine has its own effect on the cardiovascular system. So that actually brings up a very interesting point. I did not talk about the heart. So this effect of caffeine on cardiovascular system, there are actually healthy stresses on the heart. For example, when we exercise, it is interesting that studies show that the heart remodeling as a result of exercise is productive. It makes heart more efficient and better compared to heart remodeling that occurs as a result of anxiety and stress hormones. So there is a lot more to be discussed. There is a lot more to be known, but I wanted to touch upon it because I was improvising. And once again, Dr. Cyrus, he actually said that I'll join from my phone. And I thought that this is not fair. He's in the pathway of the hurricane and he didn't have internet. I'm going to invite him once more, maybe next week or the week after, and we'll go from there. And because it is improvised, because it is doodly, I'm going to hide it as well. Thank you very much. I'll just going to look at this for a couple of seconds to see if there are any questions, and then we'll close. Dr. Turab says that excellent basic science says, well done. You made it easy for an osteopathic sprain. Thank you very much. CL Hub says, I do like your explanation of the stress reaction. Yes, the link will be active. If somebody has a link, they can watch it, but I don't have it. My usual way of teaching is that I have the backup studies that I show and I have drawings and all. That's not here. Jana says that I would have some video to share. Jana, on my channel, I do not allow links. So even if you share it, that link would be removed by YouTube. So Helen says, how about a young child who has anxiety? So of course, the right way is to speak with the psychologist and psychiatrist and work with them to figure out how to resolve it. Once again, children have a very different behavior compared to our brain. So I should then clarify that this study was in people 60 and above. Children have active brain growth, active tissue growth, active replenishment. So that doesn't mean they should stay under stress. So talk with psychologists, psychiatrists to manage that stress, but they would have a much more plastic behavior of their brain. They would have a much better outcome. So I hope that this was interesting and you're very welcome, Maro. Jean says, Improviser planned, always enjoyable and great. Thank you so much. Chrissy says, very interesting. So I completely bypassed the effect on the blood-brain barrier, the effect on the heart, and then the effect on the baroreceptors. These steroids actually affect the baroreceptor system as well, which further put a lot of problem with the heart and cause incorrect blood supply. So I have left out a number of things, but wanted to kind of show you some mechanisms that are at play. And we should try to manage our anxiety as we could, as we can. Cool. So you're very welcome. Thank you very much. Like, subscribe, and share. Your comments are welcome as well. And I would see you again. Bye for now.